So last class, yesterday, we actually started to talk about cells, not in a lot of details, but we figured out where did the cells come from. How we discovered that life is actually cellular. Okay, talked about the works by Margulis, by the works by Semmelweis and Lister in establishing the idea that infections are actually caused by microbes. And next step for us will be to overview the main components of the bacterial cell. There are many species of microbes, many species of bacteria. Okay, many species of bacteria, many species of archaea, and they have different cellular structure actually. Okay, they do have differences. And this picture shows you the kind of the overview of the prokaryotic cell. I wanted to make a little digression. We are quite used to the name prokaryotes, right? Prokaryotes are the organisms that do not have nucleus, correct? But if we think about the tree of life, the tree of life that was developed by Woese and Fox, the three domains of life, you may recollect that we have bacteria, we have archaea, and we have eukarya, right? And two of them are closer to each other, eukaryotes and archaea. Does that make sense? So from the standpoint of classification, actually, term prokaryotes doesn't really reflect the relationships between the species. Does that make sense? You see what I'm trying to say here? When we say prokaryotes, we put together two very, very different groups of organisms that they look similar. It's like if you look at the whale and you look at, I don't know, the shark, they look a lot similar, but they belong to completely different classes of organisms, right? Does that make sense? Same here. And in many cases, the most strict biologists, they insist on ditching the term prokaryotes. But if you think about that, using that word just to describe the cellular structure is quite convenient, to be honest. I mean, we say prokaryotes because they just don't have nucleus. They have certain structural features, right? So we're going to use that word. I'm using that word here and there just to highlight that in the particular case, we talk about the cell that does not have nucleus. So going back to the structure, what can the typical prokaryotic cell, what can it have? Which parts? Here on the picture, you can see different cellular organelles. Well, it's not organelles, structural parts of the prokaryotic cell, the word function next to it. It is to help you to quickly describe what each part does, because I will ask you about this. You will have questions like that on the quiz or on the exam. Okay, so it's sort of a training opportunity here. So let's take a quick overview. You have cytoplasm, which function is what? Storage, fill up the cell, store things. You have ribosomes. What do? Ribosomes make proteins, very good. You have nucleoid, which is essentially what? DNA, storage of genetic information. Different inclusions. What do? Inclusions. Like bubbles. Huh? Store, yes, store stuff. Plasmids. Ever heard about this? Let's leave it enigmatic, okay? We'll figure out what plasmid does. We have flagellum, movement. We have pilus. Surprisingly not, we'll figure it out. We have capsule. Protection. We have cell wall. Structure, yeah. Plasma membrane goes in the same category, right? And we have fimbriae. What do they do? Attachment, yes. So we have some structures that you may haven't heard about, and our task is to talk about them and figure out what those structures do in prokaryotic cell. So before we move on to the structures, let's talk about the shape of bacterial or archaeal cells. Why it is so important. If you would look at human cells, they can assume human cells or any type of mammalian cells. They often assume different shapes depending on the growth conditions. You look at, I don't know, fibroblasts, they have one shape. You look at hepatocytes, they have different shape, okay? Bacteria, it turns out, have fairly restricted number of possible shapes that their cells can assume. And this shape thing helps us to classify bacteria and, as a consequence, to identify them when we look at the bacterial cells under the microscope. So there are several categories, subcategories. So we'll start with the round-shaped bacterial cells, and they are called cocci. Thank you. So cocci, coccus, in Greek means grape. They shape like a grape berries. Okay. Of course, you'll know cocci, streptococci, staphylococci. Indeed, if you would look at Streptococcus or Staphylococcus cells, they're going to be round-shaped. Another very common shape is rod shape that is called bacillus, and the plural is bacilli. Okay, shape of the cell may again be reflected in the name of the species. Bacillus anthracis, Bacillus subtilis. Okay, cell that is shaped like a curved rod is called a vibrio. And again, the name of a genus, the genus Vibrio, Vibrio cholerae, causative agent of cholera, is named Vibrio because it assumes the shape of the curved rod. Okay. Now those are three big categories. They sometimes can combine in a shape of Coccobacillus. Okay. So you see small. So it's somewhere in between the round-shaped coccus and rod-shaped bacillus. There are two shapes that are spiral, the spirillum, and spirochete. The spirochete is longer, and usually it looks thinner. Spirochete that you have heard about are the causative agents of syphilis, Treponema pallidum. Spirillum, if you've heard about Lyme disease, you've heard about the bacteria that is shaped like spirillum. Borrelia burgdorferi, the causative agent of Lyme disease, is a spirillum. Okay. The problem with this bacteria, with the spirillum and spirochete, is that they are fairly thin, and it's hard to identify them under the light microscope because of the size, using regular bright field microscopy. Spirillum and spirochetes are usually observed in the dark field. So you have to know the basic shapes of the microbes. Okay, this. So cell shapes. Some microbes are, if you look under the microscope, and if, so, should I start with, okay. When you make a wet mount, and you collect a little bit of the microbial culture, and you mix it into the water, okay, if you make diluted enough mixture, you may observe individual cells. Does that make sense? However, sometimes cells deliberately form certain associations, okay, certain arrangements. And those arrangements are sometimes reflected in the names of the microbes. For instance, well, some of them are, some of them aren't. For instance, there's a microbe called Micrococcus luteus. It is a bacteria that lives in the skin. Micrococcus often forms shapes of two cells called diplococci. You can actually look at the smear, the wet mount, and see a very distinctive pattern of two cells arranged together here and there. Right? So diplococci, two cells of a coccus. Tetrad, 4. Okay. Now, some microbes form long chains, like Streptococcus, and actually microbial species like Streptococcus pyogenes, causative agent of the disease that you know is a strep throat. Cells of Streptococcus pyogenes, cells of Streptococcus pneumonia, they form long chains of spherical cells. Staphylococci, species like Staphylococcus aureus, form big clusters, bunches of cells. And in fact, when we're going to do the microscopy lab and you're going to do the wet mount of various microbial species and look at them, you will see that Staphylococcus indeed forms the clusters. Okay? Does that make sense so far? Bacillus may also form cells that are shaped like rods, may form long chains, and in this case, chains of bacilli are called streptobacillus. Does that make sense? So what you have to know about this? You have to understand what the Diplococcus is, what the tetrad is, strepto and staphylococci, why we call them strepto, chains, why we call them staphylo, bunch. Actually, in Greek, staphylococcus is the bunch of grapes. Okay. Streptobacilli, why are we called streptobacilli? Because chains of bacilli. Does that make sense? Okay. That's pure memorization. I mean, there's probably some logic if you know ancient Greek, but I don't think you do. How cell, bacterial cell, maintains its structure. The environment in which cell lives is usually cell. What is it surrounded with usually? Water. Some sort of a watery environment. Whether it's, I don't know, soil, there's usually a thin film of water. Soil is wet, right? Whether it's a body, an organism in which bacteria lives, so on. That makes sense? And cytoplasm of the cell also consists of water, mostly. Does that make sense? Okay? Now, water that is outside, water that is inside of the cell, they're going to move back and forth. And the process in which water crosses semi-permeable membrane, which is the cell membrane. How do we call this process? Osmosis. Very good. That's osmosis. Water in the process of osmotic movement, in which direction does it go? Towards what? Huh? Of what? Okay, so water goes from its high concentration to its low concentration. Do we talk about water concentration when we talk about solutions? Do we regularly say this solution has such and such concentration of water? Not typically. What do we say? This solution has such and such concentration of solute, whether it's sugar, a salt, a protein. Does that make sense? Low water concentration means, so you have two solutions. One has high concentration of water. One has low concentration of water. What about solute concentrations in them? It's reversed. Does that make sense? And look, here's my problem with you, all of you, understanding concentrations. That's going to be a little bit of a rant here. Everybody thinks, oh God, concentration is such a badass chemistry. It's really complicated. No, it is not. If you have two bags, each bag has apples and oranges. If one, and they have equal number of like fruits, okay? Like 30 fruits in each bag. If one bag has 20 oranges, how many apples? If another bag has 20 apples, how many oranges? Does that make sense? It's reverse. You have no problem with apples and oranges. What's the problem with water and solutes then? Think about them as apples and oranges. Those are molecules, apples and oranges. I don't really care what you name them. You can name water apples and solutes oranges. Who cares? Think about them not from the standpoint of chemistry or physics. Think about them from the standpoint of common sense. Little thingies that moving around, and it will make your life 1,000 times easier. Does that make sense? So water moves in terms of the solute concentration from low concentration of solute to high. Does that make sense? Sometimes, to make it easier to process, what I say that water always tries to dilute things. Does that make sense? It tries to dilute things. So, how it goes to the concept of osmolality and tonicity. So what is osmolality? Osmolality is the number of solute molecules, not which molecule it is, not size of the molecule. Does that make sense? So if you have a solution which consists of salt and sugar, both salt and sugar will contribute to osmolality. Does that make sense? Okay? Does that make sense? Good. Tonicity is, it describes relative amount of solutes in the solution. So if you have a hypothetical cell, okay, you have solution surrounding it, and if osmolality of both solutions is equal, how do we call these two solutions? Inside of the cell and outside of the cell. Isotonic. Iso means the same, right? Now, if cell is in the environment with very high concentration of solutes, how do we call such environment? Hypertonic. One thing that I want to highlight here. Tonicity is the relative parameter. Does that make sense? So if the environment has higher concentration of solutes than the cell, then it's hypertonic. We do not talk about absolute numbers. We talk about relative numbers. And vice versa, if the cell has higher if this, concentration of solutes outside, okay, is lower than inside of the cell, that's going to be, environment is going to be hypertonic. Not so obvious question, but bacterial cells, in most cases, in which environment they live? Isotonic, hypertonic, hypotonic. Huh? Hypo or iso. Mostly actually hypotonic environment. So in the hypotonic environment, water, in which direction does it move? Huh? Into the cell. Do we get it? So what happens to the cell? It's otherwise everything, all of them would die. No, it swells. Okay? What prevents cell from going boom? Cell wall. Membrane would not. Membrane is too soft. That's cell wall. Cell is kind of, how to say, what's the word? But it's swollen, like, like a balloon. Okay, like you pump the balloon, like a tire. It makes it shaped a certain way. Now, that's for bacteria. Take protozoa. They do not have cell wall. How would they deal with it? Cell without the wall, they will lyse. Now, think about amoeba. Amoeba can live in hypotonic environment in the water and don't die. What do they have to do with the water that gets into the cell? They have to pump it out. Exactly. This is called, oh crap, I forgot, it's a vacuole. I forgot the name of that vacuole. When I'll remember, I'll tell you. It's not important for now. There's a special vacuole that actually, so amoeba has to constantly pump out the water. Does that make sense? It takes actually, if you would look at amoebae in water, they eat all the time because they have to produce enormous amount of ATP to pump out water. So it's kind of an endless cycle. Okay. Now, we talked about the bacteria that the fact that they live mostly in hypotonic environment. If you would go to California and hop on a plane and fly around the San Francisco Bay Area, there are going to be little farms that make sea salt. Any idea how they make it? Yeah, they evaporate it. And what they do, they have like pool basins, okay? So they have a basin, and the water evaporates, and it reaches a certain concentration, then they drain it to another basin where it evaporates, farther, and they drain it to another basin, and they just step by step. And they actually have certain concentration in each basin. And you know what? Each basin has a different color. Like some pink, some yellow, some red. The reason for that? Archaea and bacteria, which can survive in actually pretty high concentrations of salt. Talk about 7, 10, 15% of salt. To give an idea what 15% of salt is, imagine having a cup of water, like 1 cup, with 30 grams, 2 tablespoons of sodium chloride in it. This is salty. But microbes can live in it. How? I'll give you a clue. I deliberately didn't say hypertonic environment. I said salty environment. Yes. they have high concentration of salt inside the cell. Does that make sense? So if the salt in the environment is 10% and salt inside of the cell is 10.1, it's hypotonic for them. Does that make sense? That's the way those, if you would put such cell into the normal environment, it would die. Some microbes can actually regulate the salt concentration. For instance, staphylococci, like Staphylococcus aureus. They can survive on the regular salt content like TSA agar. Or you can put them on the medium which contains about 5% of sodium chloride, which is pretty high. And they'll grow on it just as well, because they can immediately increase the concentration of salt inside the cell. Does that make sense? Why we talk so much about hypertonic environment? What did we discuss? How do we preserve food? We salt it, we pickle it. Because microbes don't really grow well in the salty environments. Not all of them, I mean. Does that make sense? So we create environment with very high osmolality, and most of them just can't grow. That's what happens to them, actually. Water leaves the cell and it just shrivels. Okay. Right? Finally, we made it to the discussion of different parts of the cell. Remember I told you that there are many different, we already seen many different cell shapes. Cells have different structures, and remember that picture with the bacterial cell with all this flagella, pili, blah blah blah, fimbriae. Does every cell have all of this? No. Then we need to ask ourselves another important question. Which parts does every bacterial cell have? Of course you can read from the slide, but it's no fun. Let's figure it out. Seriously, because when you are in the exam, you won't have the slide. You will have to figure it out, so let's do it now. What does every cell have? Cytoplasm. Without cytoplasm, there is no cell, right? So we have cytoplasm. What else? Membrane. Does it, go ahead. It's not either or. It's a membrane. Membrane is the must. If there's no membrane, there's no cell. I mean, it's just, there's no boundary. Okay? It's membrane. Some reason I don't have it here, but let's put it membrane. Okay. We're going to have it farther ahead. Okay, membrane. What else? Nucleoid, DNA, right? Nucleoid. Nucleus will get to eukaryotic, but bacterial, prokaryotic cells must have DNA. So nucleoid. And the fourth thing. Yes, it must have ribosomes. Why? Yes. There's no ribosome. There's no protein. There's no protein, there's no function. Four things: cytoplasm, membrane, nucleoid, ribosome. Every bacterial cell that you look at will have it. Does it make sense? So this is all in the picture, so you can see the nucleoid. Now, nucleoid is not surrounded by the membrane, but it assumes certain space in the cell. Does that make sense? Like in the houses sometimes with the, like, what is called open floor plan, when you do not have strict separation between, say, kitchen and the living room, but by the assembly of the furniture, you can say, okay, here's the kitchen and here's the living room. Usually people don't put the couch in the middle of a kitchen, okay, and dining table in the middle of the living room right in front of the TV, like 2 feet from it, okay? So, same goes for nucleoid. DNA is not scattered all over the bacterial cell. It actually is in a certain place. Got it? Okay. Now, these four things are in every cell. We're going to talk more about them. Now it's time to talk about ribosomes. So ribosomes in bacteria consist of two subunits, 50S and 30S. And when you put 30S subunit and 50S subunit together, it gives you 70S ribosome. Doesn't it look weird to you? 30 plus 50 gives you 70. Well, these units, 30S unit and 50S unit, the 50S or 30S, it refers to a very specific parameter. This parameter describes how fast these parts of the ribosome can go down if you spin the tube with the ribosomes. Does that make sense? No? Yeah, so you have a small subunit, which is going to be my fist, and the large subunit, which is going to be this bottle. Okay, and imagine they are in the tubes, and you start to spin these tubes, and these things, they're going to precipitate on the bottom. Obviously, this one will go faster, right? Because it's bigger. This one will go slower, but will go also. The speed at which it goes down is that 30S or 50S. The unit S refers to the last name. You don't have to write it down. Last name of a Swedish scientist named Swedberg. Okay. Now, if you put them together, the speed at which they go down isn't only about the mass. It is also about the shape. Does that make sense? It's also about the shape. So 30 plus 50 doesn't really add up. Okay. So this is the bacterial ribosome. It's different from eukaryotic, and we'll discuss the implications of it. Another thing that can be found in many bacterial cells is called inclusions. You can see various examples of inclusions in these pictures. Inclusions are mostly used as the storage of nutrients. I want to ask you, bacteria, do they live in the conditions of abundant nutrients? Do they starve? Or do they live somewhere in between? Most of them. Most of them. You know why? Why not abundant? Some of them really starve, like really bad, but most of them live in between. You know why? Imagine that you're all hungry. And I will bring here, I don't know, 27? Three large pizzas from Pizza Hut. That would be barely enough slice per person. So, I mean, you wouldn't be filled up, okay? You would quench your hunger, but it's not abundance. It's not that you're going to be starving, but you're not going to be fed up, to this. Does that make sense? Because of what? Because of competition. I would actually watch the fight for the last slice. Okay. In your guts, you have 1,500 species of bacteria. I'm talking about the ones that we know about. It's probably more. In terms of the cell numbers, it is estimated, and estimations are wildly different, from 10 to 100 trillion bacterial cells. It is really a lot. And they all compete for food. And food is limited there, because you eat only so much, and bacteria cannot use everything, otherwise you would die if they would eat everything that you eat. You would starve. So it's fierce competition. In some environments, bacteria really hungry, okay? So-called oligotrophic environments. But So microbes, if there is a sudden abundance of nutrients, they will store it for, lean times. They will store it in the form of inclusions. Just little, it's not even a bubble, just accumulation of chemicals, like crystals of something in the bacterial cytoplasm. Another interesting use of inclusions is storage of iron in the form of so-called magnetosomes. So imagine you have like clumps of iron inside of the cell and you know what this bacteria can do? They can orient themselves along the lines of Earth's magnetic field. You can actually isolate so-called magnetotactic bacteria from Earth using a very simple device. Huh? Absolutely. You sample of soil, throw it in water, add its magnetic stir, and make it move really slow. Bacteria will just start sticking to it. That can be done. They will follow the magnet because the magnetic field around that magnetic stir is going to be stronger than the Earth's magnetic field. Why they do it? Why they follow the magnetic field? We have no clue. It's enigma. We don't know. It's pretty cool. There's a little typo. It's endospores, of course. Now, endospores is quite a unique structure that can be found in bacteria. I mentioned them before, they are escape pods. If bacteria ends up in an environment that has no water, no nutrients, high temperature, radiation, some microbes can form endospores. The most known endospore formers are two genera, the genus of Bacilli and genus of Clostridia. So among Bacilli, you may have heard about Bacillus anthracis, the causative agent of anthrax. Yesterday, I mentioned Bacillus cereus, causative agent of food poisoning. Okay, Clostridia, of course, you know Clostridium difficile. You may have heard about Clostridium tetani, causative agent of tetanus, and now all over the news. Have you heard about California, the gas station, nacho cheese? No? One person dead ate in intensive care after they ate nacho cheese from gas station in Northern California due to the infection with Clostridium botulinum, which makes botulism toxin, arguably the most powerful toxin known to humans. And it's really bad because it causes flaccid paralysis of practically everything, including diaphragm, all muscles, including diaphragm, so respiratory arrest and death. It's really quick, and there was contamination of I don't know what, and it comes from the environment. So somebody handled this with really dirty hands, and there you go. Person has botulism. So how could it happen? So let's talk about spores. So how are they formed? Cell, when it is exposed to the harsh environments, it quickly replicates its own genetic information and goes into the forespore. Okay, forms so-called forespore. Forespore is a precursor of the endospore. In the forespore, DNA of bacteria is surrounded by one or several membranes, and eventually bacteria forms so-called cortex, which is finally surrounded by the protein shield. So think about this. It's a DNA, only DNA, nothing else, just DNA, packaged in the several layers of protective coating. Membranes, cortex, which consists of carbohydrates and proteins, and finally protein coat on the outside. Like an escape pod, like a safe. Does that make sense? And then since the environment is harsh, cell dies. Spore doesn't. And spore can persist in soil or in the environment for thousands of years, really. There were examples of bacterial spores isolated from amber, and when they were placed in the proper conditions, the bacteria started to grow back again. So this process of spore formation is called sporulation. In this room we have a perfect example of such dry conditions. If you will swipe tops of the cabinets, there are tons of bacterial spores there. You put it on the petri dish, you will see very characteristic growth, mostly of different bacillus species, spore formers. Now why? What happens to the spore when it gets onto the petri dish? Agar contains what? Water and nutrients. Water and nutrients stimulates the process of germination. Okay? Endospore germinates into the vegetative cell. I am the person that is as far from planting as possible. Is that the same word, germination? Is that the same word that is used for seeds? Okay, so seeds germinate, and then the spores germinate. Seeds germinate to make eventually a plant, and then the spores germinate to make a bacteria. Does that make sense? Okay. So what you have to know about this, I don't really care about cortex, coat, but you absolutely must understand. If we expose certain bacteria to the harsh conditions, they will sporulate, forming endospore. You will then take this endospore and put it in the appropriate conditions with water and nutrients. Endospore will germinate and produce vegetative cell. When we say vegetative cell, it is a cell that can reproduce. Endospores cannot. Endospore cannot reproduce. If one endospore, it stays one endospore. Does that make sense? Okay, good. So this is actually an example of some bacillus species with a bunch of endospores. You see those blue thingies, blue dots? That's an endospore. That's the picture of the endospore by itself. Okay? So these cells are forming spores. While this cell to your left, okay, it's not a cell actually. This is the endospore. Now, why we talk so much about endospores? Well, first of all, you just saw what happened. I mean, I just told you what happened in California, right? Most likely, the contamination came from the endospores. How else can a person acquire botulism? Have you heard? If you can food, like do your own. If you don't sterilize it well, don't handle it properly, and Clostridium botulinum gets into the can, it's a perfect anaerobic environment, and it's anaerobic. So what it starts to do, it starts to replicate there in anaerobic environment. Endospores germinate, okay? Clostridium starts to So you now have vegetative cells, they reproduce, and they will produce gas, hydrogen. And if the lid of your can is popping up, throw it away. Just throw it away. Because at this point, it's the weapon of mass destruction. Really, it's as toxic as it can be. Okay. So, endospores, have you heard about tetanus? When you get tetanus shot? After what? Like a wound, yes. I mean, not deep cut that you make with a sterile knife in the surgery room. You go outside, you play in the woods, you get a deep cut. Why? Spores, endospores of Clostridium tetani are all over in the soil. So technically, it's not a bacteria that wants, well, quote unquote, wants. Bacteria don't want anything. Wants to kill you, just so it happens that you get it. And it gets into the body, oh, that's a perfect environment. A lot of nutrients, a lot of water, I'll start to replicate, says it. And, you're screwed unless you get a tetanus shot. Does that make sense? Spores, the problem with them, they're really hard to kill. Let me put it this way. If you boil it, it doesn't kill endospores. Like regular boiling, no. There are some ways, pretty much autoclave kills everything. But I don't assume we have autoclaves at home. So, membrane. Cell membrane consists, as all you know, you should be tired at this point hearing this information. It consists of phospholipid bilayer, okay? Which means you have phospholipid molecules with the hydrophilic heads and hydrophobic tails, which make a membrane. The surface of the membrane that faces extracellular fluid or cytoplasm is hydrophilic, but inside the membrane is hydrophobic. And that is what determining the transport across the membrane, right? So how transport can happen? Which types of transport across the membrane do you know? Okay, active transport and so protein facilitated transport. So active transport. What's so special about active transport? Requires energy in the form of ATP. It's actually pumps, right? This is the example of sodium-potassium pump. Okay, so it moves, but in terms of the direction of ionic movement, in active transport, ions move from low to high. I usually illustrate it with this picture. So if you have some kind of a slide and you need to, I don't know, roll a ball like Sisyphus from the bottom of the slide, which is right here, to the top of the slide, you will use some energy, right? Just pushing it up. Does that make sense? So that's active transport. It requires energy. Any other way to get things across the membrane? Can you roll it down? Sure. How do we call it? Huh? Passive transport, or from high to low, diffusion. Now we'll talk about what is facilitated, what is not. Facilitated is when something helps you. It's going to be proteins. Transmembrane proteins, like this one, okay? Transmembrane protein is like a door, okay? It can be a channel, it can be a carrier, but the point is channels and carriers like these guys, they help certain molecules to diffuse across the membrane. Now, another type of diffusion is simple. Those molecules are like David Copperfield. They can go directly through the membrane. Okay, they don't need any facilitation. Which molecules can cross the membrane directly without any help. Diffuse through the membrane. They should, lipid soluble, right? Because the inner part of the membrane is fat, is lipid. Does that make sense? Now, which molecules then use transmembrane proteins? Water soluble, like ions. Sure. So simple diffusion is when water soluble, in simple diffusion, small lipid soluble molecules, lipid soluble, like ethanol, I don't know, fats, small fatty acids, can cross the membrane. They can cross directly. Facilitated diffusion is intended for the transport of water-soluble molecules like ions, glucose, fructose, sugars, different sugars, amino acids. They are water-soluble. They cannot cross the membrane directly. Does that stick? Now, what you need to know: structure of the membrane, phospholipid bilayer, which part of the membrane hydrophilic, which part is hydrophobic, and which molecules what is the difference between diffusion and active transport? Diffusion is along the gradient, active transport is against the gradient. And finally, which molecules use which type of transport? Simple diffusion for lipid-soluble, facilitated diffusion for water-soluble. Active transport is to move from low to high concentration. Does that make sense? So we're going to discuss cell wall, and then we're going to take a break. Cell wall is something that certainly a unique feature of the microbes, of the bacteria actually. Not all bacteria have cell wall. Okay? Not all of them. But those that have can be divided, most of them, majority of them can be divided into categories: gram-positive and gram-negative. And we're going to figure out what is the difference between them. But before we, move to the differences, we need to figure out what are the similarities. So what is cell wall made of? It is made of the polymer. It is called peptidoglycan. So peptido refers to which building blocks? Peptides are made of, proteins are made of smallest building blocks, amino acids. So peptides, peptido part, refers to amino acids. Does that make sense? Glycan refers to what? Sugar. So amino acids and sugars. The structure is actually very interesting. So in these pictures, showing you gram-positive and gram-negative cell wall, you can see the sugars represented as orange and blue spheres. Okay? They form long chains, glycan chains. Are we okay with that? Do you see that? Long glycan chains. Those are two sugars called N-acetylglucosamine and N-acetylmuramic acid. You don't have to know the names. It is fine. It's not a chemistry class. But you have two different types of sugar molecules, carbohydrate molecules. Okay? And they form chains. Now, if you try to make a fabric, if you just take threads and line them one next to another, you're not going to get any fabric. Do you agree with me? It's not going to work. You need to link those threads. And that's where the amino acids come into play. Amino acids form the peptide linkages. I'm actually going to highlight it with the green. You see those linkages? Does that make sense? You follow me? So you have chains of glycans, chains of sugars linked by peptides. Essentially, it creates some kind of a mesh. Does that make sense? That's your cell wall. Mesh. Chains of glycans linked by peptides. Right? We got it. What if you... So, cell wall has to be made. Cell makes, actually, its wall, okay? It synthesizes it. What if you will start to mess around with the cell wall synthesis? What if you will inhibit it? What's going to happen to the cell? There's no cell wall. What's going to happen to the cell? There is no cell wall. And dies, yeah, it lyses. A lot of antibacterial drugs, such as penicillin or vancomycin, interfere with the cell wall synthesis. And they are very effective in killing the bacterial cells. Does that make sense? If your cell wall isn't of a proper structure, it's easy to break it. It will just not keep the integrity of the cell. Okay? Another example of antibacterial, so lysozyme. Have you heard about the protein called lysozyme? Who took AP? Okay, you took AP. Where can you find lysozyme? Saliva, tears, snot. Lysozyme breaks down the peptidoglycan, destroys the cells. I mean, it's not awfully effective, but somewhat. Okay. Now, let's go back. So, before we move on to gram-positive, gram-negative, do we understand the structure? So it's a mesh created by the carbohydrate chains linked by the peptides made of amino acids. Okay, now, what is the difference between gram-positive and gram-negative bacteria in terms of the cell envelope? Let's take a glance on the gram-positive. So you see cytoplasm at the bottom, okay, in the left part. You see the plasma membrane, and then the outermost layer of the cell is the cell wall. So essentially, if you think about the structure that envelops the gram-positive cell, it's the plasma membrane and the cell wall. Two layers. Does that make sense? So gram-positive cell envelope contains two layers. Cell membrane and cell wall. Cell wall in gram-positive cells is thick, really thick. And it contains molecules that are shown here as this blue vertical lines, molecules called teichoic acid. Teichoic acid plays many roles. It's the virulence factor, so it increases adhesion and infectivity of the cells. It sometimes facilitates the transport across the cell wall. It regulates the rigidity of the cell wall. Okay? So teichoic acid, what is important, it is unique to gram-positive cells. Gram-negative microbes have different structure of the cell envelope. They do have cytoplasm, of course. They do have cell membrane, but then they have fairly thin cell wall, much thinner than it is in gram-positive cells. Okay? Does that make sense? And on the outside, they have additional outer membrane. Okay? Now, outer membrane is attached to the cell wall via lipoprotein called murin. And it's interesting, there's a little periplasmic space here. Cell wall is separated from the plasma membrane by the periplasmic space. One of the key elements of the outer membrane of the gram-negative cells is lipopolysaccharide. In this image, it is represented by these vertical lines that consist of the red part, pink part, and black part. Okay? Lipopolysaccharide. In the outer membrane, there are proteins called porins that regulate transport of things across the membrane. Now, why I focus so much on lipopolysaccharide? Lipopolysaccharide is one of the key virulence factors for gram-negative microbes. Okay? It is complex. You don't need to know the detailed structure of lipopolysaccharide. I'm not going to ask you these parts, okay? All you have to understand is that lipopolysaccharide means there's a lipid part of the molecule and there's a sugar part of the molecule. So lipopolysaccharide consists of fat and sugar, right? Why it's so important? It causes general symptoms of infection. When you have strep throat, how do you feel? I mean, except for crappy, I get it. Can you describe it in a little bit more details? And you have sore throat. That's why it is called strep throat. What else? Nausea, vomiting, fever. You feel lousy, okay? And you know what? This is what lipopolysaccharide may do to you, okay? Especially if the infection is caused not by the streptococci, but some gram-negative microbe. Like if you have an infection with Haemophilus, okay? Gram-negative microbes, and they die. They disassemble. Where does lipopolysaccharide, LPS, where does it go? To the bloodstream. And your immune system starts to respond to it. And it responds with generalized, non-specific symptoms. If you would look at the symptoms of different infections with gram-negative microbes, they all have this component. Okay? If you will take isolated lipopolysaccharide and inject it into the mouse, mouse will have symptoms of infection. It's going to develop fever, it's going to develop inflammation in the site of injection, general inflammation. Now, a little bit of a... So, it was pretty fundamental, wasn't it? Like lipopolysaccharide, gram-positive cell wall, gram-negative cell wall. Now, before we review that, we're going to do a little, brief draw, and I suggest you do the same. But why lipopolysaccharide is so clinically relevant? Imagine that you have some sort of IV injection you have to administer to a patient. Say you just received a very expensive drug, and it's powder, just can with a powder. So how can you inject powder in the vein? You have reconstituted in what? Isotonic solution, like 0.9 sodium chloride, right? I mean, it has to be isotonic. So how are you going to reconstitute it? well, I mean, isotonic plus powder, you just mix it, and just shoot it in. So again, well, draw it up and then just inject. Well, there's powder. Is that sterile? Well, maybe not. You don't really know. I mean, with a patient, you shouldn't try, right? You shouldn't think, it's probably sterile, it's going to be okay. This guy's going to be fine. All he has is just multiple heart failure and cancer and something else. He's going to be okay, right? So what you have to do with this? What you have to do with this solution? Sterilize it somehow. How can you sterilize the solution without destroying it? Can you boil it? You probably shouldn't. Autoclave? No. Add like chlorine to it? Sounds like a lousy idea too. Can you filter it? Oh yeah, there are filters that remove bacteria. So you take this solution, you take filter, it's 0.22 nanometer filter. You filter it through, you've got your sterile, believe me, it's going to be a sterile solution. Now you do everything sterile, you collect it in the syringe, stick it in the vein, you run the drug into the vein, and 30 minutes later, your patient develops like 105 fever. What happened? When you filter the, well, they may, but usually allergy doesn't manifest with a skyrocketing fever. What can happen? When you sterilize, you remove bacterial cells, but you do not remove lipopolysaccharide that may be in the powder, okay? As the, when cells die, lipopolysaccharide stays. It's actually pretty stable, okay? So cells are gone, but LPS is there. And the amount of lipopolysaccharide that is needed to induce that fever response, pyrogenic response, is actually very low. That is why now every drug that is IV has to go through the so-called pyrogenicity test. Okay? It has to be tested for the presence of lipopolysaccharide itself. Does that make sense? There are two ways. One is like it's not chemistry, but biological, like on the bench. Another is brutal, old-fashioned. You inject the drug, the sample of the drug into the rabbit, and take like this thermometer, stick it into the rabbit's butt, and measure temperature. Both work pretty well. Well, I exaggerated with the size. It's smaller. Don't be scared. And it's pretty thin, so it's not about the land, it's about, and you don't have to stick it like 1 foot in. So you measure, there are actually special machines that do it. You have like 25 rabbit cages, and all you have to do is put the thermometers in, and the computer will take the reads and will give you the idea. So it removes some bias, okay? And they both work pretty well. Now, before we move on to mycobacteria and then the break, what I want to do is to review with you the cell structure. Let's just draw it, okay? So, which component of the cell envelope, both gram-positive and gram-negative cells have? Mandatory. All cells, all bacterial cells have this component. Cell membrane. We got it? We're cool? In gram-positive, what is on the outside of the cell membrane? Cell wall. That's it. You see, I drew it thick, okay? and I'm going to put that it consists of peptidoglycan. And you have the molecules of teichoic acid going through it. Okay? I mean, you can see from that picture that our teacher was very generous giving me B, in middle school. But that serves the purpose. What about gram-negative? Question? What, TA? It's teichoic acid. I'm gonna. Okay? Good? So what about gram-negative? What goes outside of the cell membrane? Huh? Cell wall. Okay. Again, cell wall. Is it thick or thin? Thin cell wall. There's periplasmic space right here. So cell wall. I'm not going to put peptidoglycan in because that's kind of implied. They have similar structures. What's outside of the cell wall? Additional membranes called outer membrane. And this outer membrane, which two important components does it have? Lipopolysaccharide, LPS, and certain type of a protein, proteins. We call them porins. Porins facilitate the transport. Does that make sense? I would love to remove this, but it's just the microscope. I don't. I will try to do it actually later. We got it? Okay. Do you want me to keep it there for some time? Everybody finished this amazing drawing? Let's say a few words about other types of the cell wall, and we will take a break. Okay, so other types of cell wall. Mycobacteria, Mycobacteriaceae. They have a little different structure of the outer cell wall, so they technically, structurally, they are gram-positive. But in addition, to the cell wall, they have a layer of so-called mycolic acid. That's why we call them mycobacteria. Mycolic acid is kind of a waxy thing. Okay, it's like additional waxy layer. It protects mycobacteria from phagocytosis very effectively. Okay? Does that make sense? Now, gram staining that we discussed yesterday doesn't really work with mycobacteria. That's why we detect them using so-called acid-fast staining. Acid-fast is so pretty much just different reagents, and it stains mycobacterial cells red. Okay. Some bacteria do not have cell wall at all, like mycoplasma. You may have you heard about that name, mycoplasma? No? Mycoplasma. Causative agent of walking pneumonia. Mycoplasma pneumonia. And Mycoplasma genitalium causes mycoplasmosis, the reproductive infections and urinary tract infections. So these guys, without the cell wall, they are called millicutes. Okay? Instead of firmicutes, bacteria with the cell wall. Firm, solid, firmicutes. Archaea lack peptidoglycan. They have different polymers. These polymers may be glycoproteins, maybe polysaccharides. Okay, so archaea have just chemically different cell wall. Right? So to sum it up, what do you have to know about the cell wall? First of all, what is the structure of the cell envelope for gram-positive, gram-negative? Gram-positive have 2 layers, membrane, cell wall. Gram-negatives have 3, membrane, cell wall, outer membrane. What are the distinguishing features? Teichoic acid for gram-positive, lipopolysaccharide and porin for gram-negatives. What is the difference between mycobacteria and everything else? Well, presence of mycolic acid that gives them an additional layer of protection, right? And the fact that mycoplasma, not mycobacteria, mycoplasma, do not have a layer of do not have cell wall at all. All they have is a membrane. Got it? Good. Let's take a break. When we will come back, we will do some research.